Welcome to the Combine program webinar, Mental Health and Microbes. Can your gut bacteria affect your mood? Thank you for everybody who joined us for this webinar. I think we still have people joining as we go, according to this attendee count on my computer. But I'll just introduce myself. I'm your host speaker. I'm a behavioral and molecular neuroscientist, and I'm interested in how both gut microbiota and the immune system influence brain function and also may influence mental health. My mouse lab runs in the Department of Psychiatry and Behavioral Neurosciences at McMaster University, and I'm also a research associate at the University Health Network in Toronto. I'm here in Toronto with Adam, our KT coordinator, so if you run into any trouble as we're going through this, just send us a little message through the question control panel of the GoToWebinar control panel. Much of the work that I'm going to talk to you about is supported through team grants from the Ontario Brain Institute, including my association with the Province of Ontario Neurodevelopment Disorders Network, the PON Network, and the Canadian Biomarker Integration Network in Depression, or CANBIND. So the way this will work is I'm going to do a presentation probably for about 30 minutes, maybe 35 minutes that I'm prepared, that hopefully everybody can see on your screen. And then if you should have a question, you could type it in at any time into the control panel on the GoToWebinar control panel. And then other than my voice, which you will hear throughout, everyone else is muted. And this is just to do with maintaining the ability of everybody to hear and to ensure that the audio is sort of coming through well. So if there's any problems on your end, the best way for you to communicate with us is to type a question or a comment into the question comment bar. So we'll get started. Okay, so today I'm going to talk to you about how microbes influence mood, but first I want to just provide a bit of background. So the gut-brain axis is a bidirectional communication system. Communication along this axis is continually active and is essential to maintaining good health. There are many ways that the brain and the gut communicate. Neural pathways exist, including the autonomic nervous system and the enteric nervous system. Humoral or blood-borne pathways also are mediated by pituitary and gut hormones that can be released either from the brain, from your peripheral tissues, from your gut, and by immune signaling mediators such as cytokines that are released into the blood. Immune cells themselves can in fact communicate between the gut-brain axis and are important communicators, including monocytes or lymphocytes. As well, metabolites, including short-chain fatty acids, as well as neuroactive molecules, are key signaling molecules. And all of these systems are influenced by commensal bacteria that are residing in our gut. So the idea of gut-brain communication influencing health is not a new idea. And in fact, if we look around, reference to the gut and emotion can be easily found in our everyday language. For example, it's good to be gutsy, it's gut-wrenching, your gut feelings. So these are all sort of regularly used phrases that suggest this connection between our gut and our brain is not that novel. But what is new and noteworthy is the attention that's now placed on the importance of microbiota to health and disease. And this is evident in these covers that I'm showing here on the screen in the popular press and captured in recent journal covers, including top scientific publications such as Science or Nature. But possibly more impressive is when Microbes and the Man end up on the cover of The Economist. And while garnering the public and the media's attention is part of the current trend in microbiota research, it is also notable that evidence in the advancement of our understanding of how the microbiota's role in health is also present in the literature. And so seen on this graph, the number of publications that might use the phrase microbiota or microflora is steadily increasing. However, at this time, the number of reviews in this literature significantly outnumbers the number of primary papers, highlighting the need for more work in this area. So why the emergence of this new sort of area of microbiota in the brain. So as often the case in science, recent advancement in this area were possible due to the advances in sequencing technology and metagenomics. 10 years ago, identification of commensal bacteria was limited since the only tools available were culturing samples, and many of the species that we now know are present in the gut were not culturable. Sequencing and related metagenomics tools and bioinformatics are now readily available. And so using these tools, researchers have been able to determine the composition and diversity of bacteria due to the presence of 16S ribosomal RNA genes that is uniquely found in bacteria. So we sequence the DNA we isolate from bacteria, and as such, this 16S ribosomal RNA acts as a fingerprint for different species of bacteria, so that we can use these tools both to get the composition of the bacteria in the gut, but also the diversity as to how much different bacteria might be present. So how important are these microbiota? Consider the following. Based on recent estimates, there are as many bacteria in your gut as there are cells in your body. Well, this says that we are part microbe and part human. If we consider the genetic material as well, we are far more microbial than we are human. So this is shown in this schematic on the bottom left, where our human genome contains approximately 23,000 genes, whereas the human microbiome contains millions of genes. And so if we consider the genetic load, we're actually more microbe than we are human. And if we take it a step further and consider what we know about our genetics and individual differences in genetics, from the Human Genome Project, we actually learned that we share approximately 99% of our DNA with our neighbors. So our friends and neighbors and I and ourselves are 99% similar at the level of DNA. In contrast, by best estimates, inter-individual similarity in bacterial DNA is closer to 10%. So perhaps our bacteria is more representative of who we are than our DNA is. So what do we know about microbiota? So the gastrointestinal tract of an adult human contains 100 trillion viable bacteria. More than 1,000 species can be represented in healthy individuals. There's very great variability, as noted in my previous slide, between individuals. But what's interesting is that the gut microbiota profile within the same individual tested over time shows minimal differences. So that if I tested your gut bacteria of your own samples today, and again in 6 months, they would look very similar. Our host genetics and our microbiota or ourselves have a symbiotic relationship, and what we know is that microbiota are essential for host pathogen defense, and in this way, what happens is the good bacteria fight off the bad bacteria. They're important in metabolism and nutrient uptake. For example, there's a lot of plant polysaccharides that we wouldn't be able to digest without the help of our microbes. And importantly to this talk and to my area of research, microbiota are central to brain development. And so when we think about the bacteria in our gut, we like to think about it from the perspective that it might actually be a representation of both our own DNA, but also what we've been exposed to over life in the environment, because the profile or the composition of the microbiota is influenced by genetics, age, sex, and diet. And so it can change in response to large changes, and potentially it might be useful in identifying differences that occur in subpopulations of individuals with mental illness. What we also know is that the exposure to microbes and colonization occurs at birth through the trip down the vaginal tract and continues over development. In healthy infants, as shown in this diagram on the bottom, dynamic changes in the composition of the microbiota occur during the first year of life, and these are influenced remarkably by the mode of delivery and by the diet, whether it be breastfed or bottle-fed milk. Interestingly, breast milk contains some oligosaccharides that are not available through other sources, and these oligosaccharides contribute to the composition that develops in a baby's intestinal tract. But by 1 year of age, there's a full complement of bacteria, and by 3 years of age, they have somewhat of what looks similar to what could be a young adult composition. So our topic today is microbes and mood, and in the past few years, evidence has accumulated showing that microbiota influence brain function and behavior, including aspects of mental health. Much of the progress in this area has come from animal studies, and so in order to review some of the recent clinical literature, I'm first going to show you some of the animal work and then return to people and talk about some of the clinical implications. To start, I would just like to make the point that when we look across the literature and the experiments, particularly in the regions of gut-brain research and microbiota brain research, that similar observations that have been noted in both rodents and humans related to the influence of microbiota on stress circuitry and emotional behaviors has a lot of similar observations have been observed there, and this suggests that aspects of these processes are conserved and therefore can be studied in these animal models. So while a lot of the literature to date is in animal studies, we do anticipate that a lot of this is translatable to the human situation. So in rodents, including mice and rats predominantly, we can manipulate the microbiota in several different ways. And so my lab and other groups use a variety of approaches to ask the question of whether or not microbiota influence brain function and behavior. So these are listed here in some ways, such that diet or stress, housing, probiotics, antibiotics, all of these different manipulations that we can control in the lab environment, we can then look at the impact on behavior and brain function in order to start to tease apart whether or not microbiota influence brain function and behavior, and then in the long run to tease apart the mechanisms by which microbiota signal the brain. So the first work I'm going to tell you about looks at modifying the housing conditions, and in this case, this work is done in the germ-free mouse. So this is a mouse model that was established in 1957, and these mice are generated through C-section in a sterile environment such that they then live in a sterile or gnotobiotic environment for their whole life, and they have no exposure to any bacteria of any sort or any other pathogens. And so on the bottom here we see this picture of the sterile isolator, which is one of the units at McMaster University where we keep our germ-free mice. And so what really triggered the research in the area of microbiota brain and stress research was a key paper that came out in 2004 by Nobusudo's lab from Japan. And what they showed is that when they compared normally housed mice, which in the graph on the bottom is referred to as SPF or specific pathogen free, to germ-free mice in response to a restraint stress and sampled their blood, what they observed is normally the mouse does have an elevation of stress hormones, including adrenocorticotropic hormone and corticosterone, but in the germ-free mice, they had this exaggerated hypothalamic pituitary adrenal system response and showed elevated levels of these stress hormones 30 minutes after being restrained. And so this was really a remarkable observation and started to get neuroscientists interested in the topic of microbiota brain structure. So in my lab, the first experiments that we ran, as shown in this slide, is took these germ-free mice, and we decided to have a look at their behavior. So we removed them from the sterile isolators and immediately tested them on what's called the elevated plus maze, which is shown here in the picture. And this maze is a standard behavioral approach avoidance test in rodents, where there's an open area where you see the mouse, and then perpendicular to that are two areas which have these tall closed bodies, which are sort of a safe zone. So rodents inherently have this idea that they want to explore, but they have to avoid predators. So they have this approach avoidance conundrum, and we use it as a standard to measure anxiety-like behavior. So the more time the animal spends in the open arm, the less anxiety-like behavior they're demonstrating. And if you look in the graph at the bottom, you'll see that the blue dots are the germ-free mice, and they spent more time in the open arms of this elevated plus maze, less time in the closed arm of the elevated plus maze. And if you look at the frequency which they go out to the arm, so the closed arm activity represents how many times they run back and forth between the safe zones, and it's sort of a measure of activity, whereas the open arms is again a secondary measure of anxiety-like behavior. And so the germ-free mice went out onto the open arms more frequently, and in fact spent more time there. And so following the work we showed, this link between microbiota and anxiety-like behavior, two additional groups, Rochelle Heights and Karolinska and John Cryan's group in Cork, also showed the same observation in germ-free mice. And so this is actually a very robust finding that 3 different groups independently demonstrated the same effect, linking microbiota anxiety-like behavior. But this work, again, using the germ-free mouse, is limited in the fact that there's no bacteria at all. So while this is a useful animal model to show a link between microbiota and behavior, it doesn't actually directly show this. So the key question is whether or not the microbiota are mediating the effects observed in germ-free mice. And so the best evidence that's been published on this topic was published using a fecal transplantation study that was completed at McMaster University by Dr. Chemek Bursic and his team. This study took 2 different strains of mice, the bulb C mouse and the Swiss Webster mouse, and these mice show natural differences in behavior. So the bulb C shows high levels of anxiety-like behavior, and the Swiss Webster mouse shows low levels of anxiety. And in the upper part of this graph, what they first did using older gel technologies was to look at the composition of the gut bacteria in these mice, and they showed 2 different profiles of bacteria, suggesting in parallel that there's a difference of behavior and there's a difference in bacteria. But what they did next was they took germ-free mice, and when they took the less anxious Swiss Webster germ-free mouse and gave it a fecal transplantation from the stress-sensitive bulb C mouse, they increased the anxiety of the Swiss mouse, and then when they did the reverse, gave the germ-free bulb C mouse the Swiss Webster fecal transplantation, they actually reduced the anxiety in those mice. This really does suggest, in fact, that the microbiota is driving these differences in behavior. But the question that we really want to get to is the idea of can we actually compare mice and humans? And so here we see a phylogenetic tree of ribosomal RNA from mice and from human. And this shows that, in fact, there's similarities in the 2 profiles, but the actual specific species that are found in a mouse are different than in a human. So at the species level, they're unique. But if we go up orders of magnitude through the phylogenetic tree, we actually see that there's considerable similarity at the division level between mice and human microbiota. So this is just an important thing to consider, that if in my mouse lab I show that a particular species of bacteria is linked to a behavior, it might be a different genus or species related in that same related family in people. But importantly, in mice and humans, most of our bacteria is either in the division Bacteroides or in the Firmicutes. And so there are enough similarities between mice and humans for us to consider that about some of the recent work that implicates microbiota in mood and in mood disorders. And then I'm going to talk a little bit about probiotics, which are one of the ways that we might, target differences or problems with the microbiota in individuals with mental illness. And so to date, in the past few years, there's been a lot of reports using sequencing techniques to examine changes in bacterial compositions in different disease states. And some of the best known examples and the best documented examples are in the areas of obesity and metabolic syndrome. And so what's very interesting there is the people who do that work suggest, in fact, that they could predict obese individuals from lean individuals based on their microbiota alone. Although it's not difficult, there's a whole host of features that we can distinguish those two groups. This is, in fact, an interesting biological consideration. And so within the area of mental illness, what would be very helpful would be if we could somehow come up with biomarkers or measurable things about the biology of the individual, such as their microbiota composition, that might help us distinguish different types of depressed patients within a larger cohort and maybe tailor treatment to those individual groups. But to date, there are 2 reports that have used sequencing tools to look at the microbiota in major depression. Both of these reports showed differences in bacterial composition and diversity. The first one looked at depressed patients compared to other patients with other neurological disorders and showed some differences. The second paper also looked at healthy volunteers versus depressed patients, but really hit the mark on a couple of items, because in order to assist our understanding of how microbiota may be contributing to disease, studies really need to link the microbiota to symptoms or to clinical outcomes. And so on the next slide, what I've done is I've just summarized a few of the main points from the Jiang et al. Paper that made it very interesting. So microbiota composition in MDD patients compared with healthy volunteers showed several differences at many levels: the phyla level, the family level, even down to the genus level. What they also observed were lower levels of plasma levels of brain-derived neurotrophic factor, which is a trophic factor that's been implicated in neuroplasticity, memory, learning. It has served as a marker of depression in blood samples and has been shown to be regulated by the treatment with antidepressants. But what's interesting here is that the lower levels of brain-derived neurotrophic factor that were observed in depressed patients were associated with the relative abundance of a specific bacteria, Clostridium family of bacteria. And then another observation they made was that they saw a reduced relative abundance of Faecobacterium associated with the severity of depression symptoms as measured by the MADRS, or the Hamilton Depression Scale. And these are interesting bacteria. Both of these are butyrate-producing bacteria. And in general, people in the field of microbiota to brain communication are very interested in bacterial products and how products that might be neuroactive, such as short-chain fatty acids and butyrate, might be important to how the microbiota signal to the brain. And another interesting point would be that the Faecobacterium is one of the most abundant human commensals. It represents 5% or more in most individuals and plays a key role in intestinal homeostasis. So its reduction and its association with severity of depressive symptoms is an interesting observation if, in fact, in future studies that might be linked to other features such as barrier function or inflammatory mediators. And so this is really an interesting first finding where they've made this association with some of the clinical features of disease, and that's really critical in order for us to advance our understanding both of what the microbiota are doing, but also as to whether they're good targets for therapy. there's a couple of other things that are interesting, so I'm just going to give you a couple of examples of some of the things that I think are interesting about what's happening in the field, and then I'm going to talk to you a little bit about some evidence that probiotics might be in fact helpful. And so there's a lot of neurochemicals that are present in bacteria and been produced by bacteria, and so there's a good number of people that might consider gut bacteria as a source to manipulate the way that neurochemicals are produced that might have a positive impact on gut-to-brain signaling, or on even on the local environment that might influence who else is resident there, or how the communication might influence educating the immune system. And here's just listed a whole series of different neurochemicals, including GABA, somatostatin, dopamine, serotonin, acetylcholine. These are all neurotransmitters in the brain, but also function in all sorts of parts of the autonomic nervous system and other parts of the body. So the fact that your gut bacteria can produce these neurochemicals suggests that they might be an important signaling mechanism. To date, there's one paper that just recently came out that showed that, in fact, gut bacteria producing acetate can actually signal the CNS in response to changes in diet. And what we don't know yet is whether some of these other neurochemicals are produced at higher enough quantity to actually have an effect on signaling up to the brain. So that's still to be determined. Another great area of interest, area of gene-environment interactions, and I think this is actually the area where the biggest payoff might be in the long run for clinical effectiveness. This paper here that I highlight was produced by Ruth Lay's group, and what they examined was the bacterial composition in monozygotic and dizygotic twins, so that is people with identical DNA or people with shared parental DNA. And among other things that they looked at in this study, they showed that their host genetics influenced bacterial composition because they saw that monozygotic twins had more similar composition than dizygotic twins, but importantly, not identical composition. So they contributed approximately 45% of the composition of the microbiota to host genetics, which suggests that our DNA that we're born with drives about approximately half of the bacteria that are in our gut, but that other 50% or so is being driven by environment, which would include things like early life events, diet, age, etc. And what's a key question is, if our bacterial composition is driven in part by our host genetics and in part by the environment, which bacteria are the best targets for development as psychobiotic agents? And so that's really, I think, where the field is going. And so this has been in the news, and one of the first key papers that identified an association between gut bacteria and emotional reactivity was featured in Science a few years ago. So Emran Mayer is a leader in gut-brain research and does human imaging as well as GI function, and so he showed that composition of fermented milk product with probiotics modulates brain activity, and I'll come back to this in a minute. But that really started people thinking, and some recent headlines that capture attention are things like Psychobiotics, How Gut Bacteria Mess with Your Mind, or a recent feature in PNAS by Helen Shen that highlighted microbes on the mind, and really was a good consensus of some of the recent findings. The phrase psychobiotics was really coined, I think credit should go to John Crine and Ted Dinan from Cork, and really does capture this idea of whether or not we can in fact manipulate our bacteria to improve our minds. Again, as I said earlier, the idea of benefits of bacteria on health is not new. Eli Mechnikov, the Russian scientist who got the Nobel Prize in 1908, studied a series of people and wrote this idea, moved forward the idea that the prolongation of life in subsets of people was linked to the fermented milk that they drank. And his work inspired Minoru Sirota to investigate the connection between bacteria and good GI health. And Shiroto later became the inventor of Yakult, which was our first yogurt-like probiotic drink, which is interesting because it contained Lactobacillus casei strain in 1930, and this, in fact, is one of the, still today, prominent probiotics that you will find in your drugstore or in your yogurt. And just to show a little bit of the evidence related to this, starting again with this example of an animal study. This comes out of, in collaboration with some people at McMaster and John Cryan's group, and what they showed is if they actually feed healthy mice probiotics for 4 weeks, they can reduce the anxiety-like behavior and reduce the depressive-like behavior, which is shown up the upper panel, showing using the elevated plus maze or the 4 swim tests, which are behavioral tests for anxiety and depression. But importantly, if they actually fed mice a probiotic connected with stress, they could attenuate the stress response. And if they did looking at mechanisms of how does this probiotic signal the brain, they use vagotomy in combination with probiotic treatment, and they showed that the signaling responsible for protecting the depressive behavior in this particular study was linked to the vagus. And so this is just sort of evidence in general that probiotics influence behavior and potentially under stress conditions can attenuate the effects of stress, and some of that is neurally mediated. So if we look then to see what's been done in people, the evidence of this link is slowly emerging. So I'll just go over a couple of papers that are the ones that best represent this. So in 2011, there was a study that looked at feeding, in this case, these are all in healthy volunteers, where a combination of Lactobacillus helveticus and Bifidobacterium longum was fed. In this case, they did rats and humans, but I'm showing you the human conclusion, and they measured psychological stress using self-reports and saw a reduction across healthy individuals after a month of probiotic treatment. In the paper I referred to earlier from Emma Mayer's group, they again used a commercially available fermented milk product that had a combination treatment of probiotics to healthy women, and then did before and after imaging, and what they showed is they saw reduced activity in this widely distributed brain network 4 weeks following treatment to a negative emotion recognition task. And so this was really first the first human direct evidence of brain changes in response to that probiotic. And then more recently, in a randomized control trial, which is important if we want to actually validate the effectiveness of probiotics, following 28 days of a similar mixed composition probiotic, this group recently, again given to placebo or probiotic to healthy individuals, showed a reduction in cognitive reactivity to sad mood, specifically in the areas of aggressive and ruminative thoughts. And while this isn't, a dramatic finding, it still does demonstrate in response to this probiotic, this group of healthy individuals showed a difference that suggested positive changes in this gut-brain signaling setup. So what are the good bacteria? So to date, most of the work has been done either on a bifidobacteria or a lactobacillus, which are both readily culturable and identifiable bacteria, so they're the ones we know most about. They tend to have a beneficial effect in both rodent models and in people. And if you take a look in your grocery store or in your yogurt, you're either getting one of these or both of these along with a couple of others, but they're the predominant ones in the available commercially, and they both have demonstrated evidence. But that said, not every single bifidobacteria or not every single lactobacillus is actually going to have a positive effect. And so part of what is a challenge in the field is establishing which of these bacteria, and potentially in which combination or alone, that they will have a positive treatment effect. And who should take probiotics? So this is an important clinical question, and in some of the public work that I've done in the past, I often get a question about whether or not probiotics are a cure-all, and I'll just précis any comments I make by the fact that I'm a PhD scientist and I did not go to medical school. But nonetheless, probiotics have very few to no side effects, and so that makes them a good candidate for people to take on. They tend to, in all studies, human and mouse to date, clearly have an impact on stress circuitry and anxiety-like behavior, some depressive-like behavior. So there is some optimism and enthusiasm in those domains, but certainly to date, we don't have any good evidence to suggest we can select who should take a probiotic and who shouldn't. But certainly, there is some evidence in the medical world that probiotics have a benefit in infectious situations and in other situations. And note that, you know, from our famous Canadian Hans Sele, who did a lot of the early work in stress biology, it's not the stress that kills us, it's our reaction to it. And if you're interested in hearing a little bit of what John Crine, who's a leader in this field, is about, you could listen to his TED Med talk that you can find online, where he will tell you your state of mind is dependent on your state of gut. And then I'd like to thank all the people in my team, on the CanBind team, on the PON team, and the funding agencies that support this work. And I'd be happy, if somebody has a question, you could type it in and I will try to answer it. Okay, thanks. So we have a question in. The question is, in humans, is the variance in gut biota between people, about 10%, only due to a difference in the types of the 1,000 species or in the proportion of a similar 1,000 species? So yeah, so the question is a good one. So when we refer to 1,000 species, what I really mean there and is that we know there's at least 1,000 species that could be there, but in my GI tract, I might have 700 or 800 species, I might have 1,500 species. I think the number goes up into the thousands, and so we could at species level have very different profiles. So if we went to the phyla level or to the family level, we might look a lot more similar, but when we get down to the actual individual bacteria, that certainly is where the difference comes in. And so we're not even actually sure how many species there are. We're adding species all the time as we improve our ability to sequence these bacteria. I know in some of the work that we've done in mice, when we look at what we can sequence, we get something like 5,000 or 6,000 individual reads in a particular DNA sample. But when we sort those down, we end up with about 1,400 different operational taxonomical units, which means we can't tell what all the detail it is. So yeah, so most of the variation between us is because there's just so many different bacteria to choose from. I have another question coming in here. It says, there's a lot of evidence that the microbiome modulates the stress axis, but there also seems to be emerging evidence that stress can alter the composition of the microbiome. Can you speculate about the possible mechanisms by which psychological stress can impact the microbiome? So there is an unbelievable amount of evidence that stress alters the microbiome. So we do some of that work in my lab, where we monitor changes in bacteria in response to stress in mice. This work has also been done in response to in utero stress. There's been evidence that other early life trauma in rodent models more than anything else influences that. So there's no debate that stress can in fact modulate the composition of the bacteria, and in some cases when you treat with a probiotic, you can actually attenuate those stress deficits at the behavioral level. interestingly, in a recent review that I was working on with some other people, what we found is even if the cause of the stress, whether it be anxiety or depressive-like behavior, is not psychological in nature, but is something in the nature of a stroke or another bodily disorder, probiotics are still effective in actually alleviating the anxiety or the stress that's related to that bodily disorder. So it's possible that the microbiota and targeting the microbiota could have a positive effect in some of these comorbid disorders. So I have another question coming in that is, do you mean different microbiota may in fact influence the choice of antidepressants or other therapies in the future? And yes, I think that's one of the things that I think has some potential, is that if looking at specific profiles or compositions of bacteria in patients can be predictive of other aspects of, or can subtype patients into more homogeneous groups, that could be used in order to tailor treatment and potentially to augment classical treatment, or to suggest perhaps nutritional approaches or other approaches if in fact we can tie specific bacterial profiles to subtypes and better treatment options. And what are the better, I have another question here. Not sure if how this is all coming through on the other end, because it seems as though I'm talking continuously for an hour, which is the case. But the next question coming in is, what are the better commercially available probiotic brands? And so again, I'd have to, I did not include any conflict slides related to that. I think that there's a couple of things that I can say on this that I'm comfortable perpetuating on the web forever. One is that there is evidence that the number of bacteria that we take needs to be a significant number, and so what might be housed in your everyday yogurt might not be at the number that would be efficient to be comparable to the studies with probiotics. The probiotics, the ecologic barrier, and the other ones that I cited in the probiotic work in this talk have actually been fairly well studied in research studies, and so these combined probiotics, that whether they be liquid or in a pill form, there's several different suppliers out there. But I think that looking for a high number of bacteria counts, and some measure that the bacteria is actually alive would be, you know, sort of a good thing, although there's no clear evidence that that's a criteria. But one of the things that might be important in that area is the idea that even though you take thousands and thousands of bacteria in these probiotic drinks or in these fermented milk products, very few of them colonize. And so in some of the early studies using these mixtures, people look to see whether there's a dramatic change in the composition of the bacteria. And it actually seems that even if they pass through and exit, that during transit they have a positive effect. And so it's not clear whether they replace unhealthy bacteria that look like themselves, or whether they just have an interaction as they're passing through in transit to alter the microenvironment. And so we're not completely clear, but even sometimes in some of the rodent studies now, there is evidence that some of these probiotics do colonize to a small level. But remember, it's sort of a drop in the bucket phenomena. So the fact that it is, in fact, effective in any way is remarkable and suggests that there's really more here than we understand. And so I have another question, which is a great question. It's, is diversity a sufficient measure for microbiota health? microbiome health? And I guess the answer is to be determined, but currently the sequencing techniques of which I talked about today give you both a diversity measure, which is how many different bacteria are in your gut, and it also tells you which bacteria are there, so the composition. And so depending on, and in general, a reduction in diversity has been associated with disease states compared to healthy volunteers, although there are a few exceptions to that rule. But really, there's a big effort being launched to understand what is the healthy microbiome. And so there are differences amongst individuals and across groups. And so I think that diversity is a very easy measure. And so it might be clinically relevant because possibly we can measure it quickly and easily. But other factors will become important. For example, the microbiota refer to the bacteria themselves, but the microbiome, when we talk about the microbiome, actually also includes the DNA load that comes with those microbiota. And so whether or not your lactobacillus rhamnosus is genetically the same as my lactobacillus, the metagenomics of the system is really at the forefront of some of this work, and really an important part and a piece of the puzzle. So a great question. So we have another question here, which is, what is your gut feeling about antibiotics' impact on microbiota and mental health? And so antibiotics are very interesting, and there's not really, to date, much evidence or systematic evidence about how antibiotics might impact mental health. Certainly, antibiotics affect the microbiome, and what's interesting about that is that if you have a run of antibiotics and then you stop the antibiotics and go back, your gut bacteria goes almost within a couple of days back to where it was originally. And so is there a possibility for antibiotics to be useful? Possibly. Is it likely that early antibiotics use in life could influence brain development? Likely. There's quite a bit of work in this area ongoing. We've done some work ourselves looking at antibiotics, and one of the things that's interesting about antibiotics is it depletes the bacterial composition, but at the same time likely reduces overall inflammatory mediators and probably improves barrier function. And so there's this balance between all those different signaling systems that I was talking about that might be important to consider. But antibiotic use in childhood, or I know that if you go to when I went recently to the pharmacy to pick up an antibiotic, the pharmacist did mention probiotics, and so there's a lot of sort of co-use of probiotics at the same time as antibiotics in order to make sure that your good bacteria stay healthy during the course of antibiotics.